Hello everyone and welcome to today's webinar. My name is Sandra Appler. I'm a Syngap parent and the founder of the European Syngap organization, Lyon and Friends. We are very excited to have this webinar with our sister organization in the US, Syngap Research Fund. Our talk for today is investigating the functional single-cell biology of Syngap1 pathways. I have the pleasure to introduce today's speaker, Dr. Michael Courtney. Michael Courtney is a senior research fellow at the Turku Bioscience Center in Finland. The main focus of his work has been to develop and apply optical methods to investigate neuronal responsivity and the underlying signaling networks. His research group initially focused on the roles of the J and K and P38 cell stress signaling pathways in models of neurological diseases. His team identified novel druggability of protein scaffolding mechanisms mediating NMDA receptor-evoked pathological signaling pathways acting via P38 MAP kinase by the development of first-in-class inhibitor tool compounds targeting NOS1AP. These showed efficacy in multiple neurological disease models. Meanwhile, protein-protein interaction methods were developed to help identify additional opportunities for druggability and to facilitate compound screening. Population snapshot omic data acquired from disease models and their responsiveness to compounds demonstrated the need to quantify the dynamics of signaling networks at the single cell level. To address this, his lab developed a minimally invasive optical perturbation and measurement pipeline implemented on high-throughput automation platforms. The pipeline allows simultaneous monitoring and optogenetic control of signaling circuit dynamics in individual cells in an interacting network, as well as in situ measurements of pathway kinetics. Single-cell omic analysis algorithms can be adapting to analyze the multiplexed dynamic datasets acquired. This identifies the distinct responsiveness profiles of individual cells. This insight can help interpret the consequences of disease-associated protein and signaling network disruption and the potential druggability of specific targets, aberrant pathways, and cellular phenotypes. And now the good news for our Syngap community. His lab recently began applying this approach to investigate pathways that are either regulating or regulated by SYNGAP1, together with the impact of drugs of relevance to Syngap syndrome. One ongoing project funded by us as Lyon and Friends is to identify the endogenous RAS-CHEF that counteracts SYNGAP1 function as a potential drug target for treatment of SYNGAP1 hypofunction. Funding is also being sought for additional projects to help understand the normal and aberrant activities of wild type and missense mutants of SYNGAP1. We would also like to take this opportunity again to thank you, Michael, for your commitment to our SYNGAP1 children. We are really, really grateful for everything you do. A recorded version of this webinar will be available on the SRF website as well as on the Lyon and Friends website. By the end of this presentation, you will have the opportunity to get the answer to your questions. We would love to hear from you. Please write your questions in the Q&A. For those of you just joining us, welcome, and again, Our speaker is Dr. Michael Courtney, and his talk is investigating the functional single-cell biology of SYNGAP1 pathways. It is now my pleasure to turn things over to Dr. Courtney.
Thank you very much for that introduction, very kind introduction. I'll have to share my screen first, so let's see if I can do that. Share screen. Let's see. This one, can you see my first slide?
Yes.
Second slide now. Okay, good. Try to minimize this. Okay, great. So yeah, so I'm Michael Courtney, and we've been studying signaling pathways that SYNGAP can act on for many years, but it's only recently with support from Leon and friends that we've been able to actually get started and look at SYNGAP1 function itself using the methods that we have developed. So the work I'm going to show comes from these people in the lab, Lily Lee and Barbara Ramos, and also Afra Hack, who was with us last year, who carried out a master's project on SYNGAP1 with support from Leon and friends. Okay, so first of all, I'd like to kind of define what I mean by the title. So investigating is really to emphasize that we've really just started to look at SYNGAP1 directly. So I will present to you what it is we're trying to do. We don't really have many kind of final conclusions like this, but hopefully it will be useful anyway and will stimulate some discussion. Okay, and SYNGAP pathways, by this I'm referring to intercellular mechanisms that are influenced by SYNGAP1, and particularly the RAS-ERK signaling and calcium. And when I'm talking about single cell biology, this is usually used to refer to a recent method of single cell omics or single cell transcriptomics, where the RNA levels of every RNA species is measured in each and every cell of a sample. However, I'm meaning in the broader sense that we are trying to use methods that measure from single cells and avoid those that treat the a population of cells as a single unit, because this can be quite misleading, as I'll show. And when I refer to single cell biology, so functional single cell biology, I mean that it's the dynamic changes in signaling states that we would like to follow in single cells. Those changes that may have consequences, and we can even monitor the consequences, whereas omic type snapshots, omic methods destroy samples, so you cannot really achieve true dynamic measurements with them. Okay, and then I think the aims of our lab in general have been covered just by Sandra now, so thank you for that. But basically, we are trying to identify or study protein signaling networks that are relevant to specific diseases, and with the hope to identify sites on proteins in those networks that could be druggable and therefore could lead to some therapeutic approaches later on. In order to do this, we have to develop methods for precise measurement and manipulation, ideally on single cells. And for us, this usually means optical probes, optogenetics, and gene delivery lab automation, and also what I call high throughput perturbation microscopy. So I'll explain that a little bit later, but the idea is that high throughput microscopy is conventionally used to analyze slides and fixed samples. But what's important for our studies is to monitor dynamic changes over time and to follow them up at a single cell level. And sometimes to carry out these methods, we need to develop specialized hardware for this. So this is part of our activities. Okay, so in this talk, I will try to cover 3 separate topics, which are related to one another. And that is, first of all, the idea that RAS-CEFs may be possible targets for rebalancing RAS-ERK signaling in cases where SYNGAP1 levels are too low. In the second part, we'll take a look at the effect of statins on neuronal circuits in vitro, and what we can learn by using single cell approaches. And finally, I would say the least advanced of these 3 topics from our perspective is to determine what is the effect of mutation on the localization and function of SYNGAP protein. So we believe we have some useful tools to do this, but we have to then go on to apply them. Okay, so first of all, so work done by many labs, Rick Hugen's lab, for example, but plenty of others as well, has shown that when neurons are receiving input, excitatory input, NMDA receptors open, they let calcium into the neurons, and this engages multiple signaling pathways, and this has been the topic of our research for many years. But in the specific context of SYNGAP1, what appears to happen is that calmodulin activated protein kinase 2 is activated by the calcium, leading to phosphorylation of SYNGAP1 and loss of SYNGAP1 from the vicinity of this synaptic complex, which means that RAS at the synapse, for example, which was previously under the influence of a GAP and a GEF, an inhibitory GAP and an activating GEF, now no longer has the GAP present, so the GEF predominates, which means that the RAS gets more active, the ERK becomes active, and there's an increase in glutamate receptors being inserted into the synaptic membrane. So this is a normal process, a physiological process, that SYNGAP is regulated in this way by synaptic input, and it can lead, in some cases, to enhanced synaptic activity. But if this is too much, you can imagine too much excitability can lead to perhaps seizures, and I think this has been a topic covered with in vivo models in other seminars of the series. But it's quite easy to monitor this process by looking at the ERK pathway and the influence of lowering SYNGAP levels on the ERK pathway. This has been done many years ago by Noboru Komiyama, as you can see here, in hippocampal tissue from SYNGAP knockout mice. There's an increase in ERK activity measured by a phospho-ERK antibody, and using the same kind of antibody, Gavin Rumbauer has shown in cell cultures that there's an increase in the SYNGAP knockout. Okay, so what we put together was this kind of high-throughput pipeline whereby we can take neurons from rodents, or they can be human neurons, they can be put onto plates. We can do gene transfer, we can knock down SYNGAP1, for example, we can introduce probes, and we can have an automated system to bring plates into an imager that will go through all the wells, stimulate them, add inhibitors if needed, and make measurements. And then we get a lot of data which we can analyze later on. And you'll see how this output looks in a moment. In fact, right now. So this is the system we're dealing with. So if we, in our neurons, in our microscope, we can have neurons with lowered SYNGAP1 by using a SYNGAP RNAi approach, and you can see the immunoblotting result here. We're able to knock down SYNGAP very nicely. We can now measure the ERK activity with these beautiful probes from Sergei Rigot, which indicates the level of activity of a kinase pathway, such as the ERK pathway, simply by the level of a reporter in the nucleus compared with the cytoplasm. So we are measuring a cytoplasmic nuclear ratio here as a readout of ERK, and we can use inhibitors to show this is a genuine reporter of ERK activity in the cells. So this really does work nicely. We can measure control cells. We have a baseline level, and we can stimulate the cells, induce synaptic activity, and there's an increase, as we might expect from this model. And now if we reduce the levels of SYNGAP, we have an increased baseline, and we certainly have an increased signal in the ERK pathway after synaptic stimulation. So that's pretty clear. And what else can we do? Well, we can introduce statins. So statins, as I think we know, are used clinically in some cases in SYNGAP syndrome. So the idea is that statins, rather indirectly, I would say, reduce the activity of RAS in cells, including neurons, and therefore you'd expect them to inhibit the ERK pathway. And sure enough, we can do the same kind of experiments as I just showed in the presence of increasing amounts of statins. Sorry, the order's not quite right here, but the green one is the highest concentration of, I think it's levastatin in this case. You can see a drop in the baseline ERK signal and a drop in the stimulated ERK signal. You can plot out concentration dependencies, determine how potent any one statin is in the neuronal context here, which may not necessarily be the same as in other cells. For example, there's a strong emphasis in the statin field for the purposes of reducing cholesterol to have hepatoselective statins, which may not be relevant to SYNGAP1 syndrome, but it's what's the driving force in the statin field in general. So whether they work in neurons is a question to be addressed. So I think this is something that's easy to detect here in the cultured neuron system. Now what we can do is we can knock down SYNGAP1, and we observe this increase in ERK signal using in the gray lines here, 3 different SYNGAP1 knockdown constructs. And we can combine them with a selected level of one statin or another. So we can ask the question, are we able to normalize the ERK signal and ERK response by providing a statin in this system? And it looks pretty much that we can do that. It's not that easy to kind of pick the correct statin level in order to get a good normalization, but it's kind of possible. And how long it will be possible for, it's unclear, because we've done this 15-minute experiment. We could do an experiment later, but this is a question as well. How maintained would this normalization be? But in principle, the idea of using a statin to normalize the SYNGAP knockdown effect on ERK can be demonstrated also in cell culture, and that's quite nice. It's nothing new, perhaps, or it's nothing unexpected, but it's telling us the system that we have works. It's predicting things that we can see maybe also works clinically. So can we use it to do something new, to screen for new targets? And this is where we looked at this and we thought, okay, so a lack of SYNGAP1 in SYNGAP syndrome is leading to high levels of RAS, presumably. And this must be because there is this GEF here at the other side of the cycle, and there's too much GEF given that we don't have enough GAP. And just like there's a lot of different GAP genes, and we're only talking about one particular GAP gene, the SYNGAP1 here, there's also 30 different GEF genes in human. So we could ask which of those 30 GEFs is responsible for maintaining the balance of RAS that's regulated by SYNGAP1. It's not going to be all 30 genes. It's going to be 1 or 2. We don't know how many, but it's going to be a fairly restricted number. That's reasonable to assume that. So this is where we went to Leon and friends, and they kindly helped us by funding AfroHack to get started with our master's project to take a look at this, to build an RNAi library in AAV to be able to knock down each of those RAS-GEF genes one by one. And what we're expecting here is that most of them are not relevant. They're not affecting the ERK pathway that we're measuring in the neurons, because they're doing something else, perhaps even in different cells. So they're not relevant. Some of them may do something weird, like activate the pathway that we don't expect, and this could be for a variety of other reasons we can get into if you like. But hopefully we'll find one or more that reduce the ERK activity, and that's what we're looking for. So this is an example from one of the first experiments, and sure enough, we get this kind of pattern. We get some RNAis that reduce the ERK signal, some that increase the ERK signal, and most of them don't really do very much. So the ones here on the left side are the candidates, and this is, as I said, it's a preliminary result from one plate. We have to do replicates, and we find out what is consistent, and this is the way the project went. And with Afra's project, we ended up with a few candidates to take further, but when we moved on to checking whether those candidates actually reverse the effect of SYNGAP knockdown, rather than just having an effect on their own, we've made an assumption, which may not be correct. That is, the blue line you've seen already. There are some GEF knockdowns that reduce the ERK, which is what we're interested in, and some are having no effect or doing something else. When we add SYNGAP RNAi, we've already shown a few slides back, the ERK signal actually increases. And we've assumed that those GEF knockdowns that reduce the ERK signal are going to be those that also prevent SYNGAP RNAi from having an effect. And that's actually not necessarily the case. And the first experiment that Lily in the lab did to check this showed us that actually perhaps we've been taking too simplistic approach. Again, this is just from a single plate, just some preliminary data, but it immediately shows you what's going on, that the blue curve here and the orange curve here are actually quite similar. The orange curve is displaced upwards. This is telling us that if we add SYNGAP in the orange, we have an increased ERK activity. And it's in fact these RNAis that actually should be considered as candidates for reversing SYNGAP knockdown effects, not the ones over here. So we realized we actually had to repeat the screen all over again. So the whole project took a little bit longer than we had anticipated. But at least we had developed the RNAi panel in AAV. We applied them to the ERK assay in the rodent neurons. We were able to identify candidates, now also plus and minus SYNGAP knockdown. We have those candidates, and the next stage of the project is taking them to human-derived neurons. And for this, we have iPS cells from Gavin Rumbau and also Matthias Jung in Halle in Germany. And we have funding from Leon and friends to pursue this second part of the project. So we don't have the final answer to that. It's still ongoing, but that's the first part about the RASCAFs. So I want to take a step back now, not specifically about SYNGAP, just generally about how we investigate signaling. And we can take a very global approach. We can have a model. We can have these cultured neurons, for example. We can make a model of disease. For example, we can knock down SYNGAP1, and we can ask what happens. So the ERK activity increases. Okay, but what else happens? What other changes are there? If we would have a drug, let's say we would have a RASCAF inhibitor. We've got the right RASCAF identified, and we would put it into the system, and it normalizes the ERK signal. But what else is it doing to the system? Is it having unwanted consequences as well? So one approach is to use an omics method like transcriptomics, RNA sequencing. And when we've done this kind of approach in other projects, we notice, okay, we get a control and we get a treated sample, we get a gene list, we can compare the two gene lists, we can find out some significant differences, we can go to pathway analysis. It's a very powerful method to compare those differences with the literature and predict what pathways may have been responsible to give us the result that we're looking at. And sometimes these can give a very high statistical significance, but still we end up with a predicted mechanism that is actually quite complex, and we would like to really validate whether these players are really involved, whether things are really affected. And the complexity is such that it's actually quite challenging with normal methods to validate this. So there's another aspect which is related to this. Again, we may have the pathway, we may have a compound that's targeting that pathway, but it may affect other pathways than we want. Do we have selectivity? This is a question. But another level to that is, are all the cells really behaving all in the same way? Or are there cells doing what we want, but then there may be other cells doing something else? If we just collect all the cells into a tube and make a measurement, we might get completely the wrong idea of what's going on. We may get an average that doesn't correspond to what's actually happening. And unfortunately, the signaling field, us included, have really depended on these kind of population methods. Here's an example from a paper from us from many years back. The methods are not very sensitive. So we can measure something, but we're measuring this from maybe 10,000 to 100,000 cells. And so we can say, okay, so here we have two different conditions, two different results. There's less of this signal here than here. And then we measure something else, and there's less here than here. And we can assume that, or we like to assume that this change is because of, this change is because of this change. And that's because we tend to assume that this 10,000, 100,000 cells are actually just one big cell and everything's happening in the same way, that we have a homogeneous situation. The reality, of course, is that there's a lot of heterogeneity, and certainly among neurons and in the brain, there's considerable heterogeneity. And there may be even rare subpopulations that are giving us the same signal throughout, but only the single cell methods will tell us what's really going on. There's a little bit more to this, and that is that, of course, there's dynamics. There may be changes in one signal, and there may not be changes in another pathway or another signal, and those changes may be in different cells at different times. And to distinguish these two conditions, which are clearly different in terms of the green cell, we need a temporal resolution as well as a single cell resolution, and we need to measure multiple parameters at once. So we need three things that are typically not available in the traditional cell signaling methods. And I just want to emphasize with a couple of examples how big these differences can be. So this is a typical neuron that we study, but usually we want to study not one neuron, but multiple neurons. So if we zoom out and we look at a field of 10,000 neurons, here you see a black square, because the signal, the measurement we're making is calcium for neuronal activity, an indicator of neuronal activity. And the calcium level happens to be quite low at this one time point. And remember, there are 10,000 cells, so it's pretty crowded. There's going to be a lot of dots here. So in the colors, if I start this movie, hopefully the bandwidth accepts this and you can see what's going on. You see these kind of waves of activity moving across the screen. And these waves are composed of little dots. And the little dots are each one of them is an individual cell that contains this calcium dye that we can measure from. So the point I want to make here is that at any one time point, we might have no signal, or we might have a signal that we can measure. And if we would just take one sample or another sample, we could get totally different results. And it'll be quite confusing to us to try to understand what that means unless we realize that there's actually these temporal changes. And the measurement of any one cell does not really represent any other cell. We have to really do this measurement, single cell measurement over time. This is a really nice example I took from John Albeck's work, measuring now not calcium, but the ERK pathway, and in cancer cells. So also in cancer cells, there's a lot of this heterogeneity, and also in the ERK pathway. So depending on which time point you would sample, whether it's time point 1 or 2, you would get a completely different idea of the sensitivity of ERK to a growth factor. But in reality, the ERK pathway is being activated and inactivated repeatedly in a pulsatile fashion, and it's only by measuring the time course that we really understand what's going on. Okay, so I'm repeating myself here. We have to measure over time, measure from single cells, and measure multiple pathways from each cell. That's actually very important because it's quite common to measure time courses from single cells, but because of the limitations of methods, we often have to do a different experiment for a different pathway. And the problem is then we have no idea what happened to two different pathways in a single cell if we have to do different experiments. So we really want to measure multiple pathways from every cell. And this will become important in a moment, you'll see. So we have this pipeline I kind of showed schematically, and in reality, it looks something like this. And we have these really nice probes, some of which are based on these ones I described earlier from Sergei Rigot. We can also measure calcium. There's a number of very nice probes for calcium measurement. We can measure them all in the same cell. So what's shown here is 3 different time points of the same cell, measuring 3 different pathways, because these different readouts are different colors. There's a blue one and a green one and a red one. And we can ask the microscope, okay, take a look at the blue channel, the green channel, the red channel. All right. So at the initial time point, we have a low calcium level, and the kinase activity is active if it's in the nucleus. That means this one's relatively low and this one's a bit high. After a few minutes, you can see the calcium level has gone up. These two haven't changed very much. And then later on, this one's gone down again. This one has become more active. Sorry, I've gone the wrong way. This is an inactive kinase. This one is a bit more active, but it becomes much more active at this later time point. So by combining multiple reporters in the same cell, we can really track multiple pathways at once. So what does this look like over time? So here we're measuring calcium every second from a field of neurons. We can see some baseline spiking. We can see if we induce synaptic activity, we get much more spiking, and we can terminate that, and the cell kind of quietens down here. And we can, in the same cells, measure the ERK pathway and another pathway here. So these are averages. So how do we present the responses of individual cells? So this is shown here, where every single cell in one particular field of about 80 neurons is shown as a single thin line from left to right, indicating time. So at this vertical range, it's representing this time point 0 when we stimulated the cells. So many of them have gone blue because the calcium level's gone up. And then afterwards, you can see these green streaks. And this means that one cell shows a high ERK activity and the one next to it actually didn't, even though it was next to it and under the same conditions. So there's a huge amount of heterogeneity we can visualize or we can observe among neurons when we subject them to the same conditions. So the challenge is really how to interpret this, how to put numbers on this and get simple conclusions. So fortunately for this kind of approach, there's been a very strong development in the single cell analytical algorithm field over recent years because of the advances in technologies related to single cell transcriptomics. And I've taken this example from the Allen Institute website, whereby they've taken maybe 10 to 100,000 brain cells into a single cell transcriptomics experiment and measured the levels of all the different RNAs in every cell. And from this information, maybe 10,000 different measurements from every single cell, they've been able to categorize and cluster the properties of every cell and determine what kind of neuron they are or other cell type they are. And as a result, differentiate some hundreds of different cell types. So in our experiments, we're not measuring 10,000 different RNAs. We're measuring maybe only 3 or 4 different pathways, but we have a number of different time points. We see different time profiles. So we can actually apply this kind of methodology and discriminate a number, perhaps not hundreds, but many different behaviors. And the first time we tried to do this, we immediately saw, okay, so we have an average line here. This is plotting one pathway against another. But the 4 clusters in this example, they're actually behaving quite differently. So the shape is kind of similar to the average here, but the extent is much stronger. Whereas over here, we have another population that's behaving completely differently. And if we just take an average, we can see that. Here's another example. We've been able to use clustering to identify a population of cells that are showing a very different calcium response. And you can see the responses in the pathways are very different, probably as a result of that. And that would require further experimentation to demonstrate. But the other populations of cells, they're actually very similar in terms of calcium, and yet the 2 pathways we're measuring here are actually showing quite different outputs. Some have very big responses and some really don't respond at all in this pathway 2, for example. So most assay types provide these averages, which are really unrepresentative of individual cells, and especially in the drug discovery context, where it's actually quite common to use population averages. We would like to know which behavior we're trying to modify and whether small molecules we're testing in a screening context are actually having the effect that we want on the right population and not having some other effect on another population. And you'll see an application of this in a couple of slides. So one thing is that this is all a little bit complicated. The visualization is tricky. So I want to introduce one form of visualization. Actually, it's shown here, this kind of UMAP it's called. For those of you who are not familiar with it, but maybe you've seen it before in this seminar series. So the idea of the UMAP is that you take all the thousands of parameters that you have for every single cell and position the cell in 2D space according to the value of those parameters. So what this does is it brings similar cells together. So the clustering I did earlier was by a completely independent method, but you can see the UMAP is positioning these pink cells, these cluster 7, together in one group, maybe not completely in the same way, but pretty similarly, and the cluster 6 is together, and so on. So the advantage of this is we've put cells together according to their kind of behaviors. And now we can start to visualize, for example, where is the calcium changing? Where is the ERK changing? Where is something else changing? And I'll very briefly want to show this. This is the same UMAP projection of quite a small experiment, just a couple of hundred cells. The data of 80 of them is shown here. And I'm showing just the calcium response, which is happening in all of the cells at this time point, number 13 and 14. And a little bit later on, the calcium level has gone down, but you can see that the 2 pathways, the ERK and the other one, they're actually happening in different populations here. And this helps us visualize what's going on, but it's still quite a lot of data and it still takes a lot of analysis to really understand this. Now I want to move to statins because we've applied this to statins. So I think we know statins are very old drugs. They've been clinically used for decades now to reduce cholesterol and reduce cardiovascular risk. So we know at this stage they're really quite safe. They don't suit everybody, of course, but they're generally quite safe and can be used for relatively long periods of time. The idea to inhibit production of cholesterol by targeting so many steps upstream biochemically looks to me to be a bit indirect, I would say. And of course, there may be side effects, side consequences of this. And one of those, which is relevant to SYNGAP, is the effect on production of precursors to generate prenylated proteins. And RAS is one such prenylated protein. So RAS is also very important in cancer. So there's an interest of using statins in cancer and also in epilepsies where RAS seems to be playing a role. And SYNGAP syndrome is one such example. Okay, so we can put together a model whereby we think there's synaptic activity, calcium of course increases, we see an activation of RAS causing an activation of ERK. The RAS activity is balanced by the gap, SYNGAP1 and the GEF. If we don't have enough SYNGAP1 activity, okay, so maybe we can use a statin and reduce levels of RAS. It seems perfectly logical, so we can test this in our pipeline and see what happens. So sure enough, this I think is rosuvastatin. As we increase the level of a statin, as you've seen earlier, we reduce the level of ERK activity before and after stimulation. We can, in the very same cells, look at the calcium response. And in this kind of simplistic model, we don't seem to be trying to target the calcium, but of course we know the ERK activity leads to increased receptors at the membrane, which can increase excitability and increase calcium. So perhaps it's not surprising that we also see a trend here in reduction of calcium responsiveness or calcium levels in response to stimulation when we have put rosuvastatin into the system. As I said, we tried to measure more pathways. So the junc kinase pathway is another MAP kinase pathway similar to the ERK pathway, but there are pretty clear differences. And this we were not really expecting to change, but as you increase the statin, we actually get quite a dramatic increase in junc activity in the neurons in the same kind of concentration range that affects the ERK pathway and the calcium response. So this is just one example from a very nice paper from Julius Zhu's lab showing a summary of that particular paper, but a lot of other work as well, that MAP kinases such as ERK, but also junc kinase and P38 all regulate synaptic plasticity downstream of NMDA receptors. And so it's not only the ERK pathway that's important for connectivity and synaptic activity, but junc plays a role as well. However, junc kinase signaling does a lot of other things, as does ERK signaling, and it's traditionally associated with cell stress. So actually, this is particularly the case in neurobiology, where it's been linked to kind of negative consequences in cells, for example, degeneration of neurons and neuronal cell death. I'd like to emphasize that the results I've shown with statins are from cultured rodent neurons in vitro, and we don't know if this is also happening in vivo, even in a mouse model. But anyway, one could say that an activation of the junc signaling pathway is perhaps not necessarily what we're looking for, and it might be something to follow up. So if we now apply our single cell analysis methods, because the data I showed, it was average data with statins, but it was single cell data originally. So we still have the single cell data available to us. We can apply the single cell analysis methods. What happens here is we've piled all the data together. The cells that haven't been exposed to statins, the cells have been exposed to the maximum amount of statins and those in between. And the cells are positioned according to their behavior. So what we end up is a kind of shape like a fish. Usually we end up with these funny shapes. And I want to highlight the clusters over here, number 7, 5, and 8, and in the tail of the fish. I'll put that fish over there, and we can go through the different responses. So first of all, the calcium response. So we can plot, this is a little bit of a different plot to what we had before. We can plot the distribution of calcium signals in all the cells under baseline conditions and in response to the stimulation, with low signal on the left and high signal on the right. So not every cell is the same, so we have a distribution here, and the distribution moves to the right when the calcium increases. But you can see this purple group here, which is this cluster over here, they've stayed down at the baseline level. They haven't really responded. And we can look at the ERK pathway in the same way, and we have the cells with the lowest ERK signal at baseline, and those that haven't responded, they are the purple cluster as well. So these ones over here. On the other hand, if we look at the junc pathway, we see these other two clusters are the cell populations that have the highest junc activity, and it doesn't really matter whether we stimulate or not, they're just high. And these are actually these two other populations in the tail. Okay, so what I perhaps should have shown at the beginning is the distribution of cells among these clusters according to the level of statin. Like I said, we've just taken all our data together and put it into one pool and asked how the cell's behaving. So of course the cells exposed to statin are behaving differently. Yes, they're showing lower ERK activity, lower calcium signal, and higher junc signal. But the point is that when we increase the statin, we can see exactly when we are getting these different behaviors in the pink, turquoise, and purple clusters. And the pink and the cells that are showing the junc response that we didn't really want are not the same as the cells that are giving the ERK suppression that is what we were looking for. If there's a difference in an activity of a pathway, there could be more activation or less inactivation. We have the result that there's a change, but if we want to understand why there's a change, we have to start to look at this. So fortunately, we have already previously developed methods which enable us to use light to switch on and off pathways, and we can monitor how quickly those pathways actually turn off when we try and switch them off. And that gives us an idea of whether the activation rate is changing or the inactivation rate is changing when we're seeing a different level of activity of a pathway. So we have not applied this method to the statin results yet, but I think this could be a useful tool to understand better what the statins are actually doing in terms of the junction. But the conclusion is that we can see that the statins in our neuronal cultures definitely does reduce ERK activity and calcium responsiveness, but there is an increase in junk activity, which perhaps we don't want. And because they're happening in different cell populations, it should be possible to separate them. The mechanisms must be different. Otherwise, you'd always get both together in the same cell. So in order to take this further, we'd like to try different statins, but we need to know that they're all acting on the ERK pathway as well. And are they all doing what they're supposed to be doing? Are they all, in the SYNGAP1 case, are they all inhibiting the prenylation of proteins? So protein prenylation looks something like this. You start off with a protein with a special motif, which RAS has, and this is recognized by an enzyme which removes these amino acids and sticks, say, a lipid group on the end of the protein. And this is the GRNL group that's produced in this pathway. So if we don't have enough of this substrate, we're not going to have so much of this pathway taking place. So you can easily monitor this by replacing the RAS with a fluorescent protein. So you can take the RAS-CAX motif, stick it on a fluorescent protein, and that is what we have here in a model lab cell line. This is it's a COS cell, and it's expressing a RAS-CAX at the end of a fluorescent protein in a turquoise color here. So we can see the membrane very nicely here. We can monitor it over time, for example, but this is a COS cell. This is not a neuron, so it's maybe not what we really want to study, but at least we know that we can monitor the ability of proteins to be sent to the membrane by this RAS-CAX motif and the effect of statin on it. If we want to know what happens if we have blocked this pathway, then we end up with the soluble fluorescent protein that is not associated with the membrane. That's actually the red one or magenta one shown here. The magenta and the turquoise proteins are basically the same, with the exception of this CAX motif. So you can very clearly see a different localization when you've labeled up the membrane or you're just labeling up the volume of the cell. As I said, these are a model cell line in the lab. They're not neurons. The neurons we're working with, we want to monitor them for 24 hours, ideally, repeatedly over time. We want to measure as many neurons as possible. So instead of getting these very beautiful zooms into a couple of cells, we want to monitor 100 cells, low resolution with minimal light. So we have very grainy data, but nevertheless, we can see, for example, here is a neuron, and we see the membrane of the neuron in the soma, and so we just see a dark patch where the rest of the cell is. We just see the membrane with this fluorescent protein, RAS-CAX. And over time, with incubation with statin, we can easily see that the empty space, apparently empty space, or the space lacking this fluorescent protein, gets filled in pretty quickly because the statin is having its effect preventing the modification of the fluorescent protein. So we end up with all the proteins in the cytoplasm. So it's very easy to quantify this. So we can measure this over time. We can add a statin like levastatin. We can use different concentrations and determine a concentration dependency. We can compare different statins and see how good each one is in our cell culture neuron model system. And the next step here, which we haven't got now, is to compare different statins for the on-target and off-target effects. So now we can determine whether the statins, any statin at any concentration at any time, is having its effect on this lipid modification, which RAS has. We can look at the effect on the ERK pathway, and we can look at the effect, the off-target effect on the JNK pathway. And we can see if there are some statins that are more selective for the on-target pathway, or if this is just a property of statins and something else about the way the statins are behaving, and we would have to take another approach to selectively disrupt the ARC signaling pathway. So this is something that we'd like to continue with, but it's not. We haven't got any results to report now. Okay, for the final few minutes, I would like to bring up the least advanced part of what I'd like to say today, and that is if we can investigate the effects of mutation on the localization function of SYNGAP1 proteins. So I think there tends to be an assumption that if we have a mutated SYNGAP1, the expression is probably lower and it doesn't work, and so we have all these consequences. We can have a disease as a result of this. And I think if you have a truncation in the gene, most of the gene is missing, then clearly you cannot have a full-length SYNGAP1 protein that is functional. But if you have a single point mutation that leads to a stop codon, you can have a similar kind of problem. But many of the variants in SYNGAP1 are missense mutations, which just changes the amino acid from one to another, but the whole protein is still there. So it might be that there's still a loss of stability, but it might also be that the protein is just not in the right place, or it's not doing quite what it's supposed to be doing. And I think in many cases we don't really know what is wrong and why. And this was a really nice study published earlier this year, in which they compared 57 SYNGAP variants. Some of these are found in diseases, others were just what they call biochemical controls. So they've intentionally activated or inactivated the gap domain, for example, and they've analyzed them on different signaling pathways to find out what do these different variants actually do different from the wild type. And I think the important thing that I've highlighted from the text here from this paper is that of these 57 variants, only 5 of them exhibited protein instability. So the vast majority of them are not simply not expressed or expressed at a very low level, but there's just something wrong with their function, something else wrong. But I'd also like to highlight that this is a huge project that they carried out here, very impressive work. Of course, they made it as simple and robust as possible by using a very standard lab cell line, the human embryonic kidney cell, HEK293. We use these cells as well. They're great cells. They're really easy to work with. But if we want to understand synaptic function, they don't have synapses, so we can't really do that. So what do we need to do to understand what these SYNGAP variants are doing wrong in the neuronal context? So we always have this kind of decision-making process on our hands when we're trying to understand proteins and signaling and protein networks. We can look at the purified proteins, and this is absolutely essential for example, drug discovery and to understand what the protein is really doing. But it's not telling us anything about what's happening inside a cell. So we have to have cells. There are nice model cell lines that are easy to work with, but often if we want to look at synapses, we have to go to primary cultured neurons, or even better, if we can use iPS-derived neurons from human. Of course, we need to consider the in vivo context, and then we have rodents. So all of these levels are important. We work in our lab in the first 4 levels and through collaboration on the 5th level. So what is the problem with using with simply applying methods that work in 293 cells on neurons? This is the problem. The standard and very easy to use transient transfection methods actually do not work very well on neurons. They work great in some cell lines, particularly 293 cells, but not in neurons. And they also have these overexpression problems. You can have really a large number of gene copies per cell. So these methods are easy to use, but a bit difficult. Some labs, like Rikuna's lab, have done amazing things in the SYNGAP field with transient transfection methods. So they have their place, they can be used, and in the right hands, they can be really powerful. But it would be preferable if we could somehow use a gene transfer method that's more effective in neurons. And the AAV method seems to be the favored one, also in vivo, and also even potentially in the clinic for gene transfer. But the problem is here, the restricted size of the AAV genome. The SYNGAP gene is about 4,000 bases long, or let's say the coding sequences. The SYNGAP gene is even bigger, but we've only got about 4,500 bases of space here to play with. And we need to have not just the coding sequence, but also a promoter. Maybe we want a fluorescent tag. We need some other components. So this is really problematic. So the solution that we think we've stumbled across is a split protein method So what we're doing here is we managed to squeeze the full-length SYNGAP coding sequence into AAV, together with some very reduced other elements that are essential, and a short peptide sequence that is doing what's called fluorescence complementation. It's complementing another protein that we put in with another AAV to generate fluorescence. So what it now means is we can put a genuine SYNGAP1 sequence into a neuron very easily, into all the neurons in the culture, and monitor with fluorescence where they are and how well they express. And here's just a preliminary example with a wild type SYNGAP construct. And as you can see, we can see this kind of synaptic type localization, these puncta. We could quantify the puncta, we could measure their intensity, we could quantify the intensity of all the protein in all the cells. Sorry, bring that there. So we can get an idea of localization stability, and we can even combine this with methods I showed earlier and look at signaling in the neural environment. And potentially, we could do exactly the same thing with SYNGAP point mutants. And we've done some preliminary experiments with some coding sequences kindly provided by Noboru Komayama, and it looks like we can observe differences in the neurons with this approach. But these are early results, so we need to do more with this. But there's another thing. Localization is good. It's telling us whether the protein is at the synapse. It's not the whole story. We actually would like to know if the protein is not at the synapse, why not? What is it? Is it localizing with something? Is it binding to a protein that it really shouldn't do? And is this what causing it? Is this what is causing it to be in the wrong place? And if we can identify something like this, then we have a potential new drug target for therapeutic approaches. We could prevent SYNGAP1 from being in the wrong place and encourage it to be in the right place in the synapse. And this is a question that can be addressed by proximity labeling approaches. And again, this is compromised by how would we get SYNGAP1 into the cells together with the proximity methods. But I think the split protein complementation methods could also be applied here. So we're hoping to follow this up. We've got next door to us a really sensitive proteomics unit who's now started to get single cell proteomics working. So that shows just how sensitive their methods are. And we are developing our optogenetic methods to try to get increased temporal resolution of the proximity labeling approach, so that we can ask the question, what is SYNGAP1 interacting with under baseline conditions, and then at particular time points after a stimulation? At what point is, for example, a variant not behaving as it should be? So these are plans for the future, and hopefully we'll be able to carry them out. But I think that's all I can show right now. And just to summarize, at the beginning, I was showing that RAS activity, which definitely depends on a balance of GAP and GEF activities, so we could potentially balance or normalize ERK signaling resulting from a loss of SYNGAP1 if we could identify the corresponding GEF in neuron that's responsible for the other part of the cycle regulating RAS where SYNGAP is active. The second part of the talk was about statins, and we can certainly show a reduction in ERK and calcium responses, but we unexpectedly saw an activation of the JNK pathway, which is probably not what we want because it tends to be associated with negative consequences in neurons. And applying single cell analysis methods, this showed us that actually these effects from different populations, and this means that the mechanisms are probably different, and it should be possible to find a way to have one outcome, that is suppression of ERK calcium responses without increasing the JNK responses. And then finally, I showed something about split protein complementation methods. They can be used to determine the localization of SYNGAP1 and the level of its expression. And I hope that we can use this to take a look at sequence variants of SYNGAP1 and understand better what is going wrong with those SYNGAP1 mutants that are expressed but are not able to carry out normal SYNGAP1 function in disease. So thank you very much for your attention. What I showed is based on the work from Lily and Barbara in the lab, and Afra Hecht, who was previously in our lab, and some work from former members of the lab. And we're very grateful for contributions from Noboru Komayama, and Gavin Rumbau, Matthias Jung for the IPS cells, and also funding support from Leon and Friends for the SYNGAP1 work. And I can take questions now if there are any. Thank you.
Thank you, Dr. Courtney. That was a tour de force. We saw two. Hans has a question. So I've got a few thoughts in my head. First, I see a few friends and scientists on the attendee list. If somebody just wants to ask a question live, you want to raise a hand. I'm happy. I can allow you to talk in the webinar, and that might be more efficient because based on the depth and the complexity of the presentation, if you type out all your questions and I read them, things will get lost. So that's my opening point. If anyone wants to ask a question directly of Dr. Courtney. My second point is I saw two unpublished papers with from Lee. When are those coming out? You can answer that at your leisure. And then Pavel had asked a question, but Hans predictably has already emailed me and said, I have a question. So I'm going to let Hans talk and start there just to get us going. So Hans, you should be allowed to talk.
Great. Can you hear me? Yeah. Okay, super. Fantastic presentation. I did want to talk more about the AAV gene therapy because, you know, it's been used for a few years now. There's data, for example, in the hemophilias, and I guess I'd like to hear you talk about it more in the sense that, you know, not all enzymes are created equally, right? So a liver expressed, you know, phenylalanine hydroxylase is different. The production of factor VIII is different. And here SYNGAP it strikes me at least as a very dynamic enzyme that goes through periods potentially of higher expression, lower expression with cognitive development, with you know, an AAV delivering episomal SYNGAP replacement. How do I wrap my head around that? If you know, you're getting one vector genome per neuron, are you making a substantial impact potentially? Are there limitations? Have you guys talked about it?
So experimentally, this isn't our area at all. So there are definitely labs working in this field, and I think I've attended SYNGAP symposia, which have had talks on this topic. So probably the wrong person to ask these questions from, but certainly it's being discussed in the SYNGAP field. From my perspective, I mean, clearly SYNGAP1 is small. There are splice variants. What we've been looking at is only one of the splice variants. How you would encode multiple splice variants in that small AAV genome, that's going to be a problem. Yes, I agree with you. In vivo, as a kind of gene therapy approach, there are so many challenges there. I don't know if one genome per cell, that may, well, one's a bit limiting, but it may not be. Yeah, getting the dose right, if that's what you mean, that could be a problem. But that's the same for drugs as well, right? So all of these things, potentially there are ways one can address them, but to squeeze them all into one AAV, I think that's too much. I think that would be tricky. Now, you can also split genes among multiple AAVs, but then delivering sufficient AAVs so that you will get one of each kind of AAV together. I think these are kind of clinical AAV questions that, or at least in vivo AAV questions that it's kind of out of my field. So I think probably I shouldn't say more. The question should really be addressed by those in that area.
Thank you.
Yeah.
Okay. And then there's a question from Pavel in the Q&A. He says, will GEF balancing intervention only help the formation of new synapses, or might it rescue function of existing synapses as well?
Yeah, it should. I don't see why it should not apply to existing synapses, because so if we take a very simplistic view, we don't have a functional SYNGAP1 gene. So if we look at the kind of our picture of what a synapse looks like, what a neuron looks like, where all the proteins are, and we pull out SYNGAP1, then we have too much GEF. Every time the SYNGAP1's been pulled away, there's too much GEF in every place where the SYNGAP1 should have been, if the GEF is still there. So in that simplistic point of view, inhibiting the GEF should be beneficial. Just like, much more indirect way, using a statin or reducing the level of RAS should help. But reducing the level of RAS there is reducing the level of everything that RAS is doing. So I think statins cannot be reducing RAS levels very much. Otherwise, they would be much more toxic. Now, I said this is a very simplistic view. So what SYNGAP1 deficiency is doing to all the other proteins is still a question. I think that of course, it's been studied in a very targeted way. So there've been very nice studies looking at the increase in AMPA receptors on synapses and at the membrane in response to loss of SYNGAP1. So there are these expected effects, but there could be a whole lot of unexpected effects that are really only going to be determined by omic approaches and ideally single-cell omic approaches. Unfortunately, those single-cell omic approaches are typically transcriptomics, and it's actually the proteins that really matter. And so this is where the single-cell proteomics would be really valuable, but this is a very new methodology that's only being started in a few places now, I think.
So again, I want to invite any of the science colleagues on the call to raise their hand if they have questions or type them in. But I want to ask you another question, Dr. Courtney. Is it fair to say that you started looking at statins because you heard that parents were doing some off-label use of statins in close partnership with their neurologist, of course? But am I correct in, like, you didn't just wake up one day and say, oh, let's try statins? Like you heard that parents were using it, and so you dug in. Is that right?
Yeah, I mean, I think from a kind of let's say a cell pharmacology or biochemistry point of view, I would not have wanted to go in that direction because it just seems very non-specific. But when it's actually used clinically, then it makes sense to take a look and to see, what else is it doing? Is it really doing what it's supposed to be doing? Or let's put it in another way: is our simplified cell system giving us the results we expect? Is it feasible? Like I said, are we able to titrate and get the right amount of statin to reverse the consequences of reduced SYNGAP1? And if our model is able to do this, then maybe it's able to do other things. And so, in a sense, in a rather simplistic way, replicate what it seems to be doing in vivo.
So, thank you for doing that, because I mean, I'm certainly one of the parents who was like, wait, this could help, and my doctor was like, oh, there's no downside, safe, let's try it.
It seems to be safe.
Yeah, seems to be safe, but we still don't understand quite what's happening with the J&K pathway and long-term consequences.
I think it cannot be suppressing RAS so strongly, because RAS is important for life, basically. I mean, this is what's been such a struggle in the cancer field. It's been known for decades that RAS is a major player in cancer, and it's been really difficult to drug. And some people are coming to the conclusions that those molecules that have been developed for the RAS pathway, the RAS-ERK pathway, are really hard to use because if they really work, they'll be really toxic in the long term, right. Let's say.
Right, intriguing. And so you packed thank you for packing so much into that. I'm going to watch that at least like 3 more times, and I'll probably get a headache. But and I apologize if I missed it because I was working so hard to parse which things I should even try to understand. But what are the next steps for your lab in terms of research areas? Is it to double click on the different statins, or is it more the AAV work? Like, where is your current fascination and that of your team?
I guess I mean, in the case of statins, those drugs are available. They've been available for decades. They've been tested, and so many people. So the clinical data is all there. I have a bit of a concern. Let's say we test a bunch of clinically used statins, and we don't find one that's any different from what we've seen already. Of course, there's a lot of kind of more preliminary compounds that are out there, experimental compounds. As I said, the hepatoselectivity has been one of the things people have worked on. So that's not necessarily what we're looking for here. But there's so much effort to go from a small molecule to a drug that's safe and can be used continuously. That's a very expensive undertaking. And it's not something that we'll be able to take very far. Let's put it that way. We could certainly take a look if we would, include that in a project that we can get funded, and it would be interesting, I think, but we can only take it so far.
Right.
In the case of the AAV delivery of SYNGAP1, I think it's really nice that we can now put SYNGAP1 variants into neurons in a kind of more, it's obviously not a physiological way, but it's in a kind of milder way. The real problem with transient transfection methods is you overexpress and you get such a huge amount of the protein you're expressing that you're interested in. Of course, you can see it, but then the cell behaves in a very strange way. And we struggled for many years with the methodology I showed today, years back, trying to do the same thing without the AAV. And it really didn't work. The cells just didn't behave well at all. It was really quite toxic. So by using AAV, we're able to overcome this limitation, and we can start to look at the kind of normal, reasonable behavior of the neurons. They're doing what we expect them to do if we hadn't transferred genes to them, except that we've now manipulated specific genes, and we can monitor in a single cell manner over time these different pathways. So I think this latter part is perhaps the most interesting one, especially if we can now include some high sensitivity proteomic methods to understand what SYNGAP1 is interacting with, or what the variants of SYNGAP1 are interacting with that they shouldn't, because that opens up a whole new range of potential drug targets, basically, if we could identify what they are.
I just got it's fun doing this because I get hit on Slack and text and the chat and there's all these little things popping up on my screen. But Omid has a question and I've just given him the mic. Are you there?
Hi,
Omid?
Dr. Courtney, Omid Karkudi with Rarebase. Just had a quick question for you. How efficient is the dual AAV transduction of SYNGAP1? And have you tried that in iPS neurons? Or has the lab tried it in iPS neurons or only primary neuronal cultures? And is it, I'm not sure if you're allowed to, but if you could share the serotype that was used in that, if you know.
So we're just starting with the iPS cells. The timing with getting involved in SYNGAP projects was great because Lily Lee in our lab had just gone to a lab specializing in iPS cell derived neurons. So she's come back to us, and so she has all these methodologies under her wing that she can apply to this project. But the serotype testing is, we've got some ideas and so on from the literature. There, basically, it's something that's about to be done in the lab, but we haven't done it yet, so we can't answer that question. And then in the rodent neurons, the efficiency is something that, so I'm showing data that I've been getting from Lily quite recently, so I would have to check with her. So in general, the AAV transduction efficiencies are really high, and if it's a little bit on the low side for some reason, we can always just use a little bit more AAV. And that has worked for pretty much all of our assays. But in the specific case of the complementation approach, I would expect it not to be a problem to have the two, because we are doing this when we're doing the cell signaling measurements. We're measuring the ERK pathway and the JNK pathway and the calcium pathway. It's very easy to get the three reporters into the same cell at the same time, no problem at all. And the knockdown construct, the SYNGAP knockdown construct, and the GEF construct. It actually becomes a problem when we have 5, and we want to make sure we get a full knockdown, because then we boost up the levels of some of the RNAi constructs. But it's definitely possible to get 5 different constructs into the cells at the same time, into the neurons at the same time. But in the specific case I showed, I can't answer the question right now. I'd have to go and check with Lily.
Very much.
Okay. Any other hands? And there's also a lot of parents on this call, which is actually where the webinar series started. And I encourage parents, if you have questions, even if that was a lot of science, which I want to emphasize is great, because as parents learn more and more, we do go back and we do watch these again and we say, oh, now I get it. So this was really wonderful. Thank you. Are there any other questions from anyone in the audience? Everyone's going to be like me. They're going to go watch it 2 more times and email you, Dr. Courtney.
Okay, feel free to contact me.
Thank you very much. So the focus of your lab is drug discovery, right? If I remember that correctly. And that was
I would say cell signaling. So the drug discovery process is a really long, expensive process, and it's we can only do a little bit of the beginning of the very much at the early discovery target identification end. There's not much we can go forward into it. Actually, we can do some compound screening as a kind of tool compound level, but to actually find lead compounds, that requires quite a lot of funding to follow up. And we have one project where we're looking into that in another case, but it's tough. It requires a lot of resources.
I want to ask you one question about your Kurt Haas slide, the missense characterization, because I feel like I'm going to get questions about that from a few people. He was only looking at missense variants, right? And what in that paper?
With the paper, yeah. Well, I think they compared to a wild type.
Okay,
right.
But
yeah.
Because I think the point you emphasized, which is interesting, is only 5 of the different mutations that he looked at had protein instability, which means the other ones were misformed but not unstable. So those patients then would not be haploinsufficient. They would just maybe have other issues along the various pathways. Am I overreaching here?
That is the conclusion you could make, but you have to remember, of course, or we all have to remember that it was in these 293 cells, which are somewhat artificial. They're very convenient,
right?
Lab cell line. But anyway, it's not that the sequences are intrinsically unstable, because these are cells. They do function like cells. They're just not neurons, and there's something a little bit different about them. But I think it's something to really take seriously, that we can't just say, oh, these proteins are unstable, unless we have pretty good direct evidence that they really are unstable. So they could more likely something else is wrong.
No, because this is a thing in our community, right? 2/3 of according to our research on our own data, 2/3 of our patients have protein truncating variants, very clear haploinsufficiency. About another 20% are missense. And then, but then if you go to the variants of uncertain significance, we have a huge pool of missense mutants. And that's actually a topic of conversation is, how do we figure out, short of waiting for clinical symptoms to present and try to figure out, is it or isn't it, right? How do we figure out and characterize these missense mutations? So it's a constant conversation among those of us who think about these things. Like, how do we help figure out the two questions? How do we figure out the missense who are us? And then how will the various therapies that are being developed apply to the missense mutants? Right? Anything that would be an upregulation is kind of an open question. So that just caught my attention that you focused on that, but the heck thing is interesting. Okay, we got 2 questions pop up. Fernando Suarez asked, is the same old statins for Syngap? I don't know, Fernando, if you want to clarify that. I'm happy to let you talk. And then Olga said, is there any data to show statins may work more effectively on a specific type of Syngap mutation?
You know that, because I don't have my hands on the clinical data. I think you probably have a lot more information about that.
Right.
And who's taking statin and who's not. Actually, I don't even know, how common is the use of statins? Right.
It's up to the flexibility of the neurologist and the sense of adventure of the parent, frankly. I mean, if I can editorialize for a second, our parents are very quickly test the creativity of their neurologists, right? Our kids are on a lot of drugs. Once, assuming seizures come under some level of control, behaviors and cognition become huge challenges. And statins are purported to help in some cases. In those 2, I think there's like 2, maybe 1-page case studies in literature. This helped a little bit, right? I think one of them, if I remember, was in an older patient, and that might be an interesting data point in terms of later stages of development. It's more useful. Early stages, there's more down. Who knows? But certainly, and I'm one of them, parents who are banging their head against the wall and feel like there aren't enough options say, well, let's try this. And I tried it, and I tried it for a few months. And then actually we spoke as it was sort of a precursor to this. And I heard about J&K pathway, which I know nothing about, but you were like, not always good. And I was like, let's stop the statins and see what changes. And frankly, nothing. I didn't see a major change. I was like, well, less is more. But but I think a lot of parents have tried it. If the parent is desperately looking for more and their neurologist says yes, those are the two things that generally have to happen. But I would say at least a dozen or so, probably more, parents are doing it. Happy to be corrected if anyone's on the phone who knows better. Fernando Suarez, do you want to clarify your question? Because I don't understand it. Is the same old statins for SYNGAP1? Or if not, we can...
Maybe the same ones used for cardiovascular conditions. So I think the answer is yes.
Yeah. Rosuvastatin is the one that most people try first, right? So it's one of them.
Mike, just to clarify, the German group is doing a study on statins, and I think Marcos is here, just in case you wanna. Yeah, Marcos is here in case.
Awesome.
Something.
Marcos, you want to say something?
You wanna promote him?
Hello, Marcus.
I let him talk. Promote him. He'd have to get off and get back on.
Hello. Good morning. What we are doing here in Germany is not really a study, it's an individual, how it's named, cure trial. So we are not allowed to perform a kind of study with our patients, and we take the risk for this kind of treatment with our children, and we have here in Germany, I think about from the group of Professor Kluger, about 10 children with this treatment. My son is taking statins since February, and he's only taking statins, and we saw after a certain time improvement in the epilepsy and in the behavior and also in the sleeping. So in our case, it works very well. But I think it takes time to see if it works or not. So many of the parents has got expectations that after 2 weeks they see some effects, but it is not so. And we noted the improvement, a notable improvement about half in a year. So I think something in the brain has to be reprogrammed or reconstructed so that the process comes back to a normal level, I think.
Interesting.
So we did a quite good experience with the statins.
And is that going to be published, Marcos?
I have no idea, because this has to be asked to our researchers, Professor Dr. Kluger and Dr. Kiebel and Dr. Eschermann. So I heard they are working on it, and but I'm not completely involved in this.
Cool. Thank you for updating. That's great to know. Okay, with that, if there's no more questions, Marta, should we wrap it up? Marta's in the, Marta might be.
No, I'm here. Yeah. This was very interesting. Thank you so much. It's amazing. The last part was very amazing. I didn't expect that. Really nice. Thank you so much.
Yeah,
Thank you.
agreed. Looking forward to seeing what you guys do next, and hopefully be in touch. Sandra?
Thank you also from Austria.
Thank you for your support.
Thank you, Michael.
Okay, with that, thanks for joining, everybody. Have a good day. Thanks again for your time, Dr. Courtney, and we'll talk soon.
Bye-bye.